Guys, welcome to our notes on osmosis. Last time we took notes, we were talking about diffusion. And so now we're going to talk about a special kind of diffusion called osmosis. So remember that osmosis is a type of diffusion. And so here we can see in this comic that we are going to be working with salt molecules and with water molecules. And so here, let's remind ourselves what diffusion is. Remember that molecules, because they have kinetic energy, they bounce off of one another and they move from areas of high concentration to areas of low concentration. And so this is how a lot of molecules in our body move around. And so now let's talk about osmosis, which is a very specific type of diffusion. It is specifically and only the diffusion of water through a semi-permeable membrane. So it is the diffusion of water through a semi-permeable membrane. And so we can see here we have a tank with some water in it, and so these little blue things are going to represent water molecules. And now we are going to introduce something else into the system, but the water molecules are going to be diffusing across this, and right here, this semi-permeable membrane. And so let's look at our own tank of water. And let's first develop a couple of our own concepts. Let me get the pen. Here we go, and I'll use orange. And so if we were to pour water into this tank, what would happen? Well, so here we have a semi-permeable membrane that water can pass through. And so if I were to pour water into one side of this tank over here, it would eventually reach equilibrium. Oops. It would be a little bit more straight than that, but it would be a flat line. It would reach equilibrium. It would spread out and be equal on both sides. What we have here now is a before and after. We have two tanks. And once again, we have a semi-permeable membrane, which is only going to allow water to pass through. Water is small enough, it's a very small molecule, that it can pass through this semi-permeable membrane, or selectively permeable. We're going to use those two terms interchangeably. And so if we were to pour water in here, and we just talked about this, it's going to reach equilibrium and it's going to spread out evenly. But then what we're going to do is throw some salt into the mix. We're going to put a whole bunch of salt into this side. We're going to make it a salt water solution. And remember that a solution has a solute, and so the solute here is salt. And let's just for fun put a little bit of salt on this side, but not very much. Just throw some in there. And so we have quite a bit more salt on this side. So we definitely have an area of high concentration and an area of low concentration. And so what's going to happen? We said that only water can pass through this membrane and salt cannot. So how is it going to reach equilibrium? What's actually going to happen is there's going to be a concentration gradient. If salt can't pass through, well then water is going to pass through. The concentration gradient is this direction. And so the water is actually going to defy gravity. It's going to push up and it's going to move up the column. And it's going to increase the amount of water on this side to make it less concentrated. And the water is going to sink down on this side, and it's going to make this slightly more concentrated until they've reached equilibrium, until they are more equal solutions or equally concentrated. So let's put this concept into words. So when salt was added to the water, and only water could pass through the membrane, the water had to move down a concentration gradient to the other side to reach equilibrium again. So what I didn't put in there, and I should have put in there, is the water had to move down a concentration gradient to the other side of the tank to reach equilibrium again. Look at what this means for living things. So here I have a really poorly drawn diagram with 3 animal cells. I drew them red, so to represent red blood cells. And so we have one of these parts of this chart is going to be for an isotonic solution. And so what is isotonic? So the prefix iso means same. And so the solution on the inside of the cell is going to have the same concentration as the solution outside of the cell. And so here, what we're going to do is let's grab our draw tool, and let's use the color, let's use black. So here, we're going to draw some salt. We're going to make a solution. So remember that a solution has a solute in water. And so here, we're going to make the concentration of the solution outside of the cell the same, iso, as the concentration on the inside of the cell. Here, this one's going to be hypertonic. So here, hypertonic is when the solution on the inside is more concentrated. And so this should make sense. So hyper means if you have more energy or lots of energy. So we're going to put our hypertonic solution on the inside of the cell. And so that means we're going to have a more concentrated solution on the inside because it is a hypertonic solution on the inside of the cell. So here, so we made that really concentrated, and let's make this just a little bit concentrated. So this is definitely less concentrated. And so hypo, if you have hypothyroidism, then you have low energy, hypo. And so the solution on the inside, in this one, we're going to put the solution on the inside is going to be hypotonic, meaning there's going to be less salt. And so here, then we put more concentrated solution on the outside. And so here, let's make it quite a bit more concentrated, just to make the point. Here we go. And so that is much more concentrated than the solute on the inside. So the inside is hypotonic to the outside. It is less concentrated. And so how is water going to move through this membrane? So remember that water can pass through membranes and very, very slowly through aquaporins. And so what's going to happen? So the salt is too big. It can't pass through this membrane. Only water can. And so how is it going to reach equilibrium? Well, isotonic solution has already reached equilibrium. It is in equilibrium. And so there's going to be no net movement. There's no concentration gradient. However, let's not forget that there's always random movement. So there's going to be movement both in and out. So there is no net movement, there's no concentration gradient, but there's still movement. So hypertonic, there is going to be a concentration gradient here. And so what direction is the water going to move? How is it going to try and make it move towards equilibrium? If the salt can't move out and water has to move somewhere, well, here we have a lower concentration of water, right? We have a higher concentration of salt and lower concentration of water. So here, our net movement or our concentration gradient is going to be to move in. And that's going to make the cell get really big. It could get really big, and like we've talked about before, so let's draw it getting big, 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 big. It could actually just explode. It could rupture, and that's not good for cells. And so the net movement in is going to make the cell grow larger. And if we have a hypotonic solution, so here we have less solute on the inside and more solute on the outside, which direction is the water going to flow to reach equilibrium? Well, I'm sure you guessed it, the concentration gradient is going to be to move outwards. It's going to make the cell shrivel. And I don't know how to draw a shriveled cell. There we go. And the water is going to move out, and so the concentration gradient is going to be outwards. And so that's not good for a cell either. What is good for this animal cell is an isotonic solution. So notice in this picture that it's switched around a little bit. Here we have our isotonic solution with our red blood cell, and this is water going in and water coming out. And it is a healthy solution for that animal cell. Over here was our hypertonic solution, and so much water rushed in that the cell ruptured or it lysed. And over here is our hypotonic solution, where the cell shriveled because the water was moving outwards. And so if we look here, we have a plant cell. Things are a little bit different in a plant cell because of that really strong cell wall. So it works better for a plant cell to have a little extra solute, a little bit extra of that salt on the inside, which is going to allow water to rush in, and it's going to push out on those cell walls, and it's going to make it more turgid. And so therefore it's going to give the plant more structure. And so we don't want a flaccid cell, and we don't want a shriveled cell either for a plant cell. And so this right here, an isotonic solution, would make the plant a little bit wilted and sad, and then this would just outright kill the plant. All right, so those are the notes on osmosis, and have a great day.